Hello everyone, welcome to the channel Microbial World. In this video, we will be discussing about Clostridium tetani. Its morphology, pathogenesis, etiology, clinical manifestations, laboratory diagnosis and treatment will be discussed in detail. Clostridium tetani is the causative organism of tetanus. It is widely distributed in soil and in the intestine of humans and animals. It is recovered from a wide variety of other sources, including street and hospital dust, cotton wool, plaster of Paris, catgut, bandages, wall plaster, and clothing. Tetanus is a disease caused by a neurotoxin produced by species of Clostridium, which damage the central nervous system of the host. Okay, so this species of Clostridium, they produce a neurotoxin about which we will be discussing later. This neurotoxin, it damages the central nervous system of the host and it causes the disease tetanus. Morphology. Clostridium tetani is a gram-positive slender bacillus about 4 to 8 into 0.5 micromuum in size. It occurs singly and also occasionally in chains. The spores of Clostridium tetani are spherical, terminal, bulging, giving the bacillus a drumstick appearance. It is non-capsulated and it is motile by peritrichous flagella. That means the flagella will be uniformly distributed over the body. Pathogenesis. Clostridium tetani produces two exotoxins, tetanolysin and tetanospasmin. Tetanolysin is a heat-labile, oxygen-labile hemolysin, and it is antigenically related to the oxygen-labile hemolysins produced by Clostridium perfringens, Streptococcus pyogenes, and Streptococcus pneumoniae. So the tetanolysin, it damages the viable tissue surrounding the infection and it optimizes the condition for bacterial multiplication. Tetanospasmin, which is also called tetanus toxin, it is a heat-labile, oxygen-stable, plasmid-coated neurotoxin, and it is the tetanospasmin that is responsible for disease manifestations. So the tetanus neurotoxin, it is released upon cell lysis after bacterial growth under anaerobic conditions, for example in case of deep puncture wounds. It is antigenic and is specifically neutralized by its antitoxin. Its toxoid form is also used for vaccine preparation. So let us discuss more about tetanospasmin, that is the tetanus toxin. Tetanospasmin is an AB toxin. It is 150,000 Dalton peptide in molecular weight. By the action of enzyme endogenous protease, it is cleaved into light A chain and heavy B chain when neurotoxin is released by the cell. Both chains A and B are bonded by disulfide bond and non-covalent forces. The A chain, that is the light chain, has enzyme activity, that is zinc endopeptidase, and heavy chain, that is the B chain, consists of a carbohydrate binding domain. Its carboxyl terminal portion binds to specific sialic acid receptors, for example, polysialogangliocytes, and it also binds to glycoproteins present on motor neurons. Mechanism of tetanus toxin. Tetanus toxin binds to polysialogangliocyte receptors. So we said the heavy chain is having a carbohydrate binding domain and its carboxyl terminal portion will bind to sialic acid receptors, right? So the tetanus toxin, it binds to polysialogangliocyte receptors present on the motor nerve terminals, and that will result in toxin internalization. Once the toxin is internalized, it gets transported from the peripheral nervous system to the central nervous system by retrograde axonal transport. When the tetanus toxin reaches the inhibitory neuron terminals, it will prevent the presynaptic release of inhibitory neurotransmitters glycine and gamma aminobutyric acid. So how the tetanus toxin is able to do this? The tetanus toxin, it inactivates proteins called snare proteins. Okay. So the snare proteins, it is involved in regulating the release of inhibitory neurotransmitters gamma aminobutyric acid. So here the tetanus toxin is inactivating the snare proteins, and thereby it will prevent the release of inhibitory neurotransmitters like glycine and gamma aminobutyric acid. So the lack of these inhibitory signals to the motor neurons and also the constant release of acetylcholine to the muscle fibers will lead to irreversible contraction of the muscles, and that will result in spastic paralysis. That is, our muscles will become stiff. Etiology. What causes tetanus? Most frequently the disease occurs following injury. Puncture wounds are vulnerable and it will favor the growth of anaerobic bacillus. Rarely the disease can also occur following surgical operations. And the disease might also occur due to local suppuration such as otitis media. It is called otogenic tetanus. Tetanus is also an important complication of septic abortion. Clinical manifestations. The period of incubation of tetanus is few days to weeks. The primary wound or trauma is infected by Clostridium tetani. Tetanus is classified into three types: generalized tetanus, localized tetanus, and cephalic tetanus. In generalized tetanus, the early symptoms are drooling, sweating, and constant back spasms. It is termed as opisthotonus. Masseter muscles are involved, which causes inability to open the mouth. It is called lockjaw, and this is a very common symptom of tetanus. Another characteristic is a sardonic smile, that is because of the persistent contraction of face muscles, and this is called rhizus sardonicus. In severe cases, symptoms of tetanus involve autonomic nervous system, which will include symptoms like cardiac arrhythmias, which is irregular heartbeat, change in blood pressure, and dehydration. The spasms, it will continue to spread to other muscles, and severe contraction of other muscles will occur, which will include head, arms, legs, and back. Death is usually caused when diaphragm muscle contraction is included, due to which the patient will be unable to exhale. Localized tetanus. The disease and symptoms will remain constant to the muscular area of infection site. Cephalic tetanus. From the word itself you can understand the initial site of infection is head. Neonatal tetanus. It will involve initial infection at the site where umbilical cord is removed in newborns, which will later progress into generalized symptoms of tetanus. The mortality rate is more than 90%. Laboratory diagnosis. Specimen. Necrotic tissue may be collected from the site of injury. Microscopy. Gram staining reveals gram-positive bacilli with terminal and round spores. Drumstick appearance or tennis racket appearance. Culture. Clostridium tetani is an obligate anaerobe that grows only in the absence of oxygen. Robertson cooked meat broth. Clostridium tetani, being proteolytic, turns the meat particles black and produces a foul odor. Blood agar with polymyxin B. Clostridium tetani produces a swarming growth when incubated at 37 degrees Celsius for 24 to 48 hours under anaerobic conditions. Toxigenicity testing. In vitro hemolysis inhibition test. Clostridium tetani produces hemolysis on blood agar, which is inhibited by adding antitoxin. This test indicates the production of tetanolysin only but not tetanospasmin. In vivo mouse inoculation test. Robertson cooked meat broth with black turbid growth is injected into the root of the tail of a test mouse. The test animal will develop stiffness which begins with the tail and then progresses to involve hind limbs, trunk, forelimbs. Death occurs within 2 days. This test indicates the production of tetanospasmin. Treatment requires debridement, that is, removal of the dead skin of the primary wound. Antibiotics are used like penicillin or metronidazole to kill bacteria and reduce toxin production. Passive immunization. It neutralizes the tetanus toxin. Vaccine requires 3 doses followed by booster doses for 10 years. So this is all about Clostridium tetani. Thank you.